And we should finish glycolysis today, and we'll get to the very beginning of the citric acid cycle. This will put us maybe one lecture behind. I realize I owe you a study guide on chapter 27. I also owe you a video on exam 3 for the questions there. I will do that soon. I also owe you video lectures and notes from last lecture, so I will get to that. I got drawn into a research project Friday afternoon that I was really excited about, and then I lost track of time, and then I didn't do any of those things. So I'll get back on task. All right. So the purpose of glycolysis is to break glucose up into 2 equivalent pieces and to generate as much ATP and NADH as we can along the way. The molecule that we're going for is called pyruvate, and maybe I've drawn it for you before. If not, here it is. It's 3 carbons. You've got a carboxylate group and a ketone, and then a CH3 group. So as we go through this today, I want you to pay attention to the logic of the reactions and sort of what nature is trying to do as we go from step to step. All right, sorry about that. All right, so in the steps we talked about last time, you have the investment phase, which is where we're going to consume 2 molecules of ATP. So the first was just catalyzed by hexokinase. It's an irreversible step, and it puts a phosphate group on carbon 6, and you're trading a less stable phosphoanhydride bond in ATP for a more stable phosphoester bond in glucose 6-phosphate. The mechanism is SN2-like. It's OH group on carbon 6 attacks the gamma phosphorus on ATP. This is an irreversible reaction. It's downhill by quite a ways. The reason I'm pointing this out is because nature tightly regulates irreversible reactions, because once you've done them, you've committed to go the rest of the way. And so the irreversible steps are the ones that are going to are going to be regulated by the cell through mechanisms we're not going to talk about. But it's useful to have a sense for which reactions are irreversible and which are just sort of in equilibrium. Having a lot of equilibrium steps allows the cell to be flexible. It's good to have a large pool of materials that can go one way or another, so that if the needs of the cell change and you need to, say, make glucose instead of metabolize it, that you can do that easily. The next step was the one we ended on last time, and this is the isomerization of glucose 6-phosphate to fructose 6-phosphate. We pointed out last time how glucose 6-phosphate is an aldehyde. Fructose is exactly the same in stereochemistry, except on carbon 2, it's a ketone instead of an OH group, and on carbon 1, it's an OH group instead of an aldehyde. And we talked about how you can convert from this alpha hydroxy aldehyde into this alpha hydroxy ketone only with proton transfer steps. So we first make the enolate with the negative charge of the alpha carbon, and that enolate has a resonance structure with a double bond between carbons and a negative charge on oxygen. And that, if we protonate on oxygen, that gets us to this key intermediate, which we called an ene diol. And the reason that's a key intermediate is because it could be a carbonyl in both directions. We could treat this oxygen as the carbonyl oxygen, this is carbonyl carbon, and then this one as the alpha carbon. Or we could treat this oxygen as the carbonyl oxygen, here's carbonyl carbon, here's alpha carbon. So once we get to that ene diol intermediate, we got a choice. We can go backwards, but if instead we remove a proton on the yellow oxygen, we get an enolate, and then we can protonate that enolate on the alpha carbon to get this ketone. Now, why would you want to slide that carbonyl over one carbon? And this is, again, the chemical logic of the process. We want to split glucose into 2 equal parts. We noted last time how this is an alpha or a beta hydroxy carbonyl compound, a potential aldol product. If you attempted to break it apart via a retroaldol reaction, you would have an enolate on this side and an aldehyde on this side. But the problem is that this would be a 2-carbon enolate and a 4-carbon aldehyde. And that's not what we want. We want 2 equal in number of carbon pieces so that we don't have to have separate downstream machinery, enzymatic machinery, to deal with those 2 different products. So notice the difference between the aldehyde and the ketone. In fructose, because we have the ketone on carbon 2, this is also a beta hydroxycarbonyl compound. And if we break the bond between alpha and beta carbons via a retroaldol reaction, we get 2 equal 3-carbon pieces. And it will turn out, as we continue on, you're going to see how the 2 3-carbon pieces that we get are actually very similar to each other and easily interconverted. All right. So moving on, but actually let me just pause. Any questions you want to ask about those first couple of steps? Yeah?
Which molecule in here did you say will drop on to the next step? In step 2, like which of the forms will move on to the next step?
Which of the forms here will move on to the next step? The ketone form or the fructose 6-phosphate form? Yeah. Okay. So we have one more thing to do before we can do the retroaldol reaction, and that is we have to take fructose 6-phosphate and we have to put a phosphate group on the OH group of carbon 1. And the purpose of doing this is to make sure that both of our 3-carbon units have similar functional groups. Carbon 6 over here is going to have a phosphate group, so we need a phosphate group on the first half of the molecule. So I'm going to abbreviate ATP. I'll show you the first phosphoanhydride bond, and then the rest we'll just call R. We're using fructose 6-phosphate, and we're going to get out of this process fructose 1,6-bisphosphate. That means there are 2 phosphate groups. Man, we're struggling today. It's tougher than I ever thought it would be to talk and write at the same time. I'm absent-minded enough that Amber, my wife, is just still not really believing that I can actually pull this off. You can say, don't worry, he actually doesn't. All right. So that will install the phosphate group here, and then we'll have ADP here. The enzyme is called a kinase because it transfers a phosphate group, transfers the gamma phosphate group from ATP to fructose. So the name of the enzyme is phosphofructokinase. And this step is also irreversible and will therefore be tightly regulated. It's downhill by about 4.5 kilocalories per mole, which is basically irreversible. You don't need to memorize the delta G numbers. I'm writing them so you have a sense for whether the reaction's reversible or not, and so that later on when you learn about regulation of enzymes, you'll have a sense for why those steps need to be regulated. But a good rule of thumb in interpreting free energies is that a difference in free energy of about 1.4 kilocalories per mole translates at room temperature to about a factor of 10 in equilibrium constant. And so this is multiple factors of 10, perhaps 1,000 times favoring products versus starting materials here. So this is quite downhill. The mechanism is SN2-like. This is very much like the mechanism in the first step. We're going to remove a proton from the oxygen, then the oxygen is going to attack the gamma phosphate group and a leaving group is going to leave. We are replacing a less stable phosphoanhydride for a more stable phosphoester. Okay. Anything else on that? So with that in place, we are ready for the Retroaldol reaction. And that is step 4. What I'm going to show you for this Retroaldol reaction is a mechanism that may be simplified relative to what eukaryotic cells do. So if you later learn that there's a little bit more complications here, hopefully you'll be able to forgive me. This, the first step of this mechanism is opening of the cyclic hemiacetal. I'm not going to show you that because we've talked about that a lot, and that's a reaction that we did previously, but I will draw the open chain form of fructose 1,6 bisphosphate, and remember it's a ketone. Okay, and looking at that open chain form, you have carbonyl carbon, alpha carbon, and beta carbon. That is a beta hydroxy carbonyl compound. We can break it apart via a retroaldol reaction. This side, which I'm highlighting in purple, would be the electrophile. Because that beta carbon is attached to a hydrogen, what's over here in purple is going to be our aldehyde, and then what's over here in orange will have been our enolate. So the way the retroaldol reaction works is you will have some base, perhaps some group in the enzyme active site, remove the proton from the beta hydroxy group. As to whether we get the negatively charged oxygen as an intermediate at that stage, or whether proton transfer occurs at the same time as the next step, I don't know and isn't really important. So we're going to show all of that happening at once. Electrons kick down to form the carbon-oxygen pi bond, and then these electrons in the bond between beta and alpha carbons go to the alpha carbon. And so that breaks apart our molecule into these two equivalent parts, or rather these 3 carbon portions. I'm just going to draw them in sort of the same orientation as they're drawn to the left. Except I'm going to make mistakes and get the wrong numbers of carbon. Whoops. Okay. This molecule is called glyceraldehyde 3-phosphate. And then the other partner is still an enolate. With a negative charge here. We can pick up a proton from some source, perhaps the conjugate acid of our base. And that's going to get us to the products, which I'm going to draw up here, but I'm going to draw them in a more conventional form where you can sort of see the stereocenters. So the aldehyde that we drew below, I could draw like this with an OH group back away from us into the page, and then the phosphate group there. And then I will draw the ketone here. All right. So this is glyceraldehyde 3-phosphate, and this is dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate comes from carbons 4, 5, and 6 of fructose, right? But from here on out, we're going to just call them carbons 1, 2, and 3. Dihydroxyacetone phosphate came from carbons 1, 2, and 3 of fructose 1,6-bisphosphate, though from here on out we're going to call them 1, 2, and 3 in this direction for reasons that you'll see in just a second. So I've used equilibrium arrows here because the change in free energy is actually negligible for this reaction. So just to put a number on it, it's basically 0. The enzyme that catalyzes this reaction is called aldolase because it's a retroaldol reaction. And this sort of illustrates a tradition in enzyme naming where you name the enzyme for one direction of the reaction, but it can also catalyze the reverse reaction, especially in this case where the starting materials and the products are basically equal in free energy, which is why we don't call it a retroaldolase. Somebody first described it as an aldolase because it could catalyze the reaction of these two starting materials to fructose 1,6-bisphosphate. The other molecule is called dihydroxyacetone phosphate. And you can see that it is similar to glyceraldehyde 3-phosphate, though different, similar though different. That's really informative. Notice that glyceraldehyde 3-phosphate is an alpha hydroxyaldehyde involving carbons 1 and 2, whereas dihydroxyacetone phosphate is also alpha hydroxy, but it's an alpha hydroxy ketone involving atoms 2 and 1, respectively. That puts them in exactly the same relationship as glucose to fructose. Glucose was an alpha hydroxyaldehyde. Fructose is an alpha hydroxy ketone. And before, we interconverted glucose and fructose using this isomerase enzyme where we made an ene diol intermediate and then switched who was the carbonyl and who was the alcohol. We're going to do exactly the same thing in step 5 of glycolysis. So here is step 5. And we only need to do this with dihydroxyacetone phosphate. The enzyme that does this is called triose phosphate isomerase. But you can call it TIM. Kind of cute. The change in free energy is, again, less than 1 kilocalories per mole. So this is definitely reversible, can go backwards and forwards. The mechanism we won't draw because it's the same mechanism as we showed before for the conversion of glucose 6-phosphate into fructose 6-phosphate. In other words, you're going to tautomerize to the ene diol, and then once you get to the ene diol, you're going to tautomerize to the alpha hydroxyaldehyde. And in doing so, you will generate glyceraldehyde 3-phosphate. And this completes the investment phase of glycolysis. We are now to the point where glucose has been split into two equal molecules, two equivalents of glyceraldehyde 3-phosphate. So in what comes next, the payoff phase, you have to remember that everything from here on out happens twice. All right, questions so far? Okay, so the goal of this payoff phase is going to be to get from glyceraldehyde 3-phosphate all the way to, erase those numbers, all the way to a different 3-carbon molecule, which I'll try to draw in the same way. And this molecule is called pyruvate. And along the way, we need to recoup our ATP investment, and then we also need to make a profit. If you look at what's going to have to change as we do this, we need to turn an aldehyde into a carboxylic acid. So that looks like an oxidation reaction. And then we need to get rid of the phosphate group and turn this secondary alcohol into a ketone. And then we also need to get rid of this alcohol and replace it with a CH bond. So the changes over here almost look like we're going to do a oxidation and reduction reaction in the same molecule. And it turns out we'll be able to make those changes without using any oxidizing agents. It'll be proton transfers and moving some stuff around. Okay, so look in the next few steps about how we're going to get to high energy intermediates that can then either make NADH or ATP. Okay, so step 6 of glycolysis takes glyceraldehyde 3-phosphate and a molecule of a phosphate ion that's just found in the cytosol, and NAD+. And the reaction that we're going to do is still reversible. And that may seem unusual because we're going to increase the oxidation state of this carbon, but at the same time we're going to be reducing NAD+. And so this is sort of a coupled reaction, which overall the change in free energy is pretty small. The enzyme that does this reaction is called glyceraldehyde, I'm sick of writing that, 3-phosphate, feel free to abbreviate, dehydrogenase, because we will oxidize the aldehyde to the carboxylic acid. All right, let me show you the mechanism of this step. It's kind of interesting. You have phosphate actually act as a nucleophile to attack the aldehyde carbon, and you get a tetrahedral intermediate. So, so far that's just plain old boring carbonyl chemistry, only you haven't really seen phosphate act as a nucleophile before. The intermediate that we get looks like this. Tetrahedral intermediate with a negatively charged oxygen. Now, back in our carbonyl chapters, you learned that when you get to a tetrahedral intermediate, you look for good leaving groups. And if there are any, then the best one leaves. It's clear that phosphate could leave. You could have electrons kick down and phosphate could leave. That would just be the backward step, and for sure that happens. But notice in our desire to get to pyruvate, we want to oxidize that carbon. So ideally, we're going to want to have this hydride group leave as a leaving group. Problem is, H- is a cruddy leaving group. And that's where NAD plus comes in. The enzyme holds NAD plus very closely to the molecule shown above, with a carbon, the paracarbon, with a positive charge, so that when we kick off hydride, it is just handed off directly to NAD plus. And that gives us NADH, and this oxidized molecule, and it's not only been oxidized to the carboxylic acid, but it's a mixed phosphoanhydride. By that I mean you've got a phosphate group on one side and a carboxylic acid group on the other, but this looks like a typical anhydride. You've got two phosphate groups in it. This molecule is called 1,3-bisphosphoglycerate. Glycerate is the 3-carbon carboxylic acid, and then the 1 and the 3 tell you where the phosphate groups are. All right, so there's some recoup on our investment, because we can take NADH to the electron transport chain and use it to drive the proton gradient, and then use that proton gradient to make ATP. And we've also made a molecule that has a weak sort of high energy bond. And that's going to allow us to treat this molecule as though what I have drawn, what I have circled in gray, is a good leaving group. Notice that if this group left, it would leave as the negatively charged carboxylate, and that should be reasonable. So at this stage, we're set up to recoup our investment of ATP. I guess before I move on, questions you have about that step? Yes?
What would be left for the phosphate then? Would it just be like 3 oxygens on the phosphate, or would there be something else?
What we would do is we're going to have a nucleophile attack phosphorus. We're going to have ADP attack phosphorus, and that's going to regenerate ATP. Yep. Okay, what else? Okay. So step 7 is sometimes called substrate level phosphorylation, to distinguish it between ATP synthesis that happens in the mitochondria. What we're going to see is that 1,3-bisphosphoglycerate is going to be attacked by ADP. And remember, everything that I'm showing you here happens twice, because we started step 6 with 2 molecules of glyceraldehyde. So I'm not going to draw all of ADP. In fact, I'm just going to draw it like this. This would be the beta phosphate group. And you will have electrons on the oxygen of the phosphate of ADP attack this phosphorus here, and the leaving group leaves. And this step is actually pretty neutral in terms of free energy, which should be a little bit surprising, I guess, because you just made ATP, and that should be expensive to make in terms of energy, because ATP is a high energy molecule. But the reason this step is 0 in free energy is that you've taken this mixed phosphoanhydride and you've replaced it with just a regular phosphoanhydride, and they are similar in stability. So by getting to 1,3-bisphosphoglycerate, you've made a molecule that is as reactive as ATP. And so it's easy to make ATP from 1,3-bisphosphoglycerate. The enzyme here is named Phosphoglycerate kinase, and it's actually named for the reverse reaction. The product of the reaction I just showed you is called 3-phosphoglycerate. So the kinase forward reaction would be transferring a phosphate group from ATP to phosphoglycerate to get the mixed anhydride. That's why the enzyme's called a kinase. Of course, enzymes don't care whether the reaction's forward or reverse. They don't care what the thermodynamics are. They just lower the free energies of the transition states. And so it's perfectly okay, even though this is taking a phosphate group off and putting it back on ATP, it's totally okay for the enzyme to be called a kinase. All right, so remember our goals for this second part. We've made some profit in terms of NADH. We just made 2 molecules of ATP, which we had consumed 2 molecules of ATP previously, so we just broke even in terms of ATP molecules. And since we want to get to the 3-carbon pyruvate with a carboxylate on carbon 1, we have accomplished having the carboxylate on carbon 1. Everything from here on out is going to be about making more ATP and about rearranging the bonds here to have a ketone on carbon 2. All right, so step 8 will be a bit of a relief because I'm not actually going to ask you to learn the mechanism of step 8. It's not that hard to understand. It involves enzyme side chains acting other than just as bases and acids. And I found when I've showed students this before, it leads to an excessive amount of like angst and worry, and so it's just not worth the pain. The enzyme that catalyzes this reaction is called Phosphoglycerate Mutase, and its job is to move the phosphate group from carbon 3 to carbon 2. Except I didn't do that. Let's try that again. All right. So this is 3-phosphoglycerate, and this is 2-phosphoglycerate. And again, it's really not as bad as you think, but it involves a histidine side chain, and it's just not worth the pain. But you can think of it as a series of SN2 reactions. All right. So we moved the phosphate group over. Now, doing that converts an alpha hydroxy acid into a beta hydroxy acid. And so it should be unsurprising. Oh, sorry, I should have told you the free energy for that mutase step is, again, basically reversible. Basically no free energy change. Okay. A beta hydroxycarbonyl compound is an aldol product, and you learned earlier on this semester that aldol products sometimes go on to form the corresponding alpha-beta unsaturated carbonyl compounds via an elimination reaction. So here is carbonyl carbon, here's alpha carbon, here's beta carbon, and after the elimination reaction, we now have an alpha-beta unsaturated carbonyl compound. The enzyme that catalyzes this reaction is called enolase, for reasons that'll become clear in just a little bit. This is also a reversible step, very small free energy change. And the mechanism is basically going to be the same as the dehydration mechanism in the aldol reaction. You will remove a proton first to make an enolate, and that step is fast. And then electrons on the alpha carbon kick down and form a pi bond between alpha and beta carbons, and the OH group leaves, perhaps picking up a proton from some other source on the way. And that gives us our product, which is called phosphoenol pyruvate. Now, as we've done these past few steps, you should sort of feel the tension building. We moved a phosphate group from the far end of the molecule to closer to the carboxylic acid. And there should be some electrostatic repulsion there, right? The phosphate group has a double negative charge, -2, and then the carboxylate has a single negative charge. And so as you transfer the phosphate group from carbon 3 to carbon 2, you're increasing the tension a little bit with this repulsion. And then when you make the alpha-beta unsaturated compound, you get rid of an OH group, but now you also have something that's basically an enol. It's an enol phosphate. That's why it's called phosphoenol pyruvate. If you ignored this phosphate and replaced it with a proton, you would have an enol. And remember, enols are not stable relative to ketones. Ketones are more stable than enols because carbon-oxygen pi bonds are more stable than carbon-carbon pi bonds, because oxygen's more electronegative than carbon, and electronegativity means electrons are held closer to the nucleus, lower in energy. So you have a molecule that's first spring-loaded with this phosphate group really close to this negative charge, and then on top of that, it's captured in this less stable enol form. So this is sort of the last straw, and we're now ready for the final step of glycolysis, which is another substrate-level phosphorylation reaction. You take phosphoenol pyruvate, and you take ADP. All right, and here is the beta phosphate group of ATP. And we're basically going to do an SN2 style reaction like we've done before, where we transfer, where we have the oxygen attack the phosphorus and then the leaving group leaves. As that negatively charged we could draw a negatively charged enol intermediate, though actually it gets negatively charged enolate intermediate. It gets protonated rapidly on oxygen as the leaving group is leaving. And that will give us ATP. But we're not done yet, because the thing that left, the thing that got liberated, is this enol, which is going to tautomerize rapidly to the more stable ketone form. And this is pyruvate. So there's our profit of 2 ATP molecules. Overall, this step is catalyzed by the enzyme pyruvate kinase. Again, it's named for the backwards reaction. Interestingly, this step is also irreversible. It's very downhill in energy, which is interesting because ATP itself has pretty unstable bonds and is really high in energy. The reason this step is reversible, again, two things. You're relieving a lot of negative charge repulsion as you transfer the phosphate group from phosphoenol pyruvate to ADP. But second, after the pyruvate enol leaves, it tautomerizes in a downhill reaction to give you pyruvate. Okay, that's it for glycolysis. Okay, so pay attention to the overall logic. These last 5 steps were about recouping our investment of ATP, oxidizing to a carboxylic acid, getting functional groups in the right place, ratcheting up the tension so that we get a high energy molecule, phosphoenol pyruvate, that is actually so reactive it easily phosphorylates ADP to give you ATP regenerated. All right, questions so far? I guess if I'm going to ask for questions, I don't start talking right away, do I? That's kind of silly. So, questions? Yeah. Where does this process occur in the cell? Great question. It occurs in the cytosol. Our next effort is going to be to convert pyruvate into acetyl-CoA, which is going to go to mitochondria. Okay, what else? I guess I could answer the inevitable question, how much of this do we need to know? All of it, no. I want you to know enzyme names. I want you to know substrate structures. That means starting materials and product structures and names. And I want you to know mechanism. Knowing the enzyme names and the structure names may sound annoying to you, but frankly, that's a really, really easy problem to get right on an exam. What is this molecule? Okay, it's 3-phosphoglycerate, moving on. Without any existential crisis, right, or any deep level reasoning, you can just remember that and walk away with, 5 to 8 to 10 points or whatever. Let's see. The other question that came up in the 9 o'clock class is, what percentage of this stuff we're talking about now will be on the final exam relative to other material? The final exam is comprehensive and written by me. That was another question that was asked. I'll get to that in a second. What percentage will be previous stuff versus this last stuff? My answer was somewhere between 0 and 100% will be on this last stuff, and then the corresponding amount will be on the previous stuff. That's really useless. The problem is I haven't written it yet, so anything I would tell you now would be inaccurate. But you can expect a chunk of the exam to be, I don't know. You will need to worry about both things, both this last unit and the previous stuff. There was a question, do I use a standardized ACS exam as my final? The answer is no. I don't. I write it myself. That may be good or bad depending on what your situation is. Some of my colleagues use the standardized ACS exam because it's a good way to measure relative to a standard whether you've learned the things that you're supposed to learn in OChem. And it's also a good way for them to gauge their overall teaching effectiveness. I don't like it because some of the questions are on stuff that we haven't talked about. And I know that some students, in an effort to maximize their grade, will buy an ACS prep manual to prepare for the test, and that seems like an unwise use of financial resources, and so I just don't want to bother with that. Okay, and then what was the other question? I think that was all the practical questions. Anything else that I didn't touch that you want to ask? Yeah?
Is this exam going to be slightly longer than the other ones?
Is this exam going to be slightly longer than the other ones? Well, it's worth 250 points instead of just 200. So yeah, there probably are going to be more questions. It all depends on how I'm feeling when I write it. Sometimes I just get tired and give up. Which is sort of what happened on this most recent exam. I got to 20 and I said, good enough. All right, well, we got a couple minutes left. We could move on to Pyruvate, and what happens to that, but I think we'll pick that up next time. We're about one lecture behind, so we'll adjust the syllabus as needed, and I will see you Wednesday.